So often here on the Exam Room podcast, we talk about all of the benefits of a plant-based diet in terms of nutrition, but also here at the Physicians Committee, we have a team of researchers who work tirelessly to end cruel and unnecessary animal experiments. And so, to talk about a lot of our highlights in this past year, 2021, we welcome Kristie Sullivan. She is the Vice President for Research Policy, and that means that she really likes to read. Kristie, thank you so very much for being here.
Thanks for having me, Chuck.
This has really been a banner year. I can't think of more than a week or two that went by without getting some sort of major exciting update from your team. I mean, just before we dive into the highlights, what has this year been like for you?
Well, you might think that things would slow down with the pandemic, right? Where people are not working as much or they have other things they're worried about. But that's not the case here at the Physicians Committee. There is so much to do that we are always looking for ways to help get animals out of laboratories, to replace them with more human relevant methods. And there are still lots of opportunities for us to do that and to continue to promote good non-animal science.
Let's talk about some of the big ones from this past year. And I want to start with Wayne State University, which is, that is something that I know you guys had your sights set on for some time. And there's been a lot of movement up in Michigan with this. So what was going on at Wayne State and how have we been able to help?
Right. So Wayne State has been experimenting on dogs for a long time. And they're using these dogs in experiments that are supposed to tell us about the cardiac health of people, how we can help people who have heart issues, who might be going into heart failure or might be at risk of heart failure, and how we can help them. And we know how we can help them, and we can research new ways to help them using human relevant methods, either using patients in clinical trials and clinical studies, or observational studies, or even using donated human tissues and organs to analyze human cardiac health. But these researchers at Wayne State are using dogs. They subject them to surgeries, hook them up to treadmills, and measure their cardiac function, and the dogs die at the end of the experiments. So we have been trying to get Wayne State to stop doing these studies, and we have recently found some help in the Michigan legislature. So we have a bill in the legislature introduced that would prevent deadly, painful dog experiments in the state of Michigan. We've put up billboards all over Detroit with stories of individual dogs killed at Wayne State to raise awareness. We have a coalition of lawmakers who have written to the school's president to highlight the issue and ask them to stop doing these experiments. So we're working hard in the state of Michigan, using the legislature's power to try to prevent Wayne State from killing these dogs.
And just to be crystal clear about this, we're pushing for that, but at the same time, there are 100% non-animal, human-relevant methods that could completely replace these tests, correct?
That's right. There are lots of ways to study human cardiac health, as I've said.
And so, speaking of studying, let's talk about studying among so many people this year who you and your team work tirelessly to educate about non-animal methods here. How many scientists have you had the opportunity to kind of, as I would say, coach up when it comes to ending animal experiments this year?
Well, believe it or not, we've trained over 5,000 scientists this year in non-animal methods. So different modalities of training, certainly. We've held a number of multi-day long, 2-3 hour trainings to really get into details of the non-animal methods and how they're used. And we also hold webinars that are shorter. They include lots of people, and it's just sort of a general overview of a particular topic. One maybe silver lining of the pandemic is that everyone has gotten accustomed to these online webinar programs like Zoom, and we have been able to use that to our advantage and reach people not only in the United States, but in Europe, in South America, in Africa, so that people who are studying toxicology for a living, either they're students learning how to be toxicologists, or they're already into their career. Maybe they're someone at a company trying to study chemicals and their effects on or potential effects on human health or the environment, or they're a regulator at the Environmental Protection Agency. All of these folks need to learn the latest. They need to stay up to date on the best human relevant non-animal toxicology methods, and we provide them an opportunity to do that. That's free, easily available. They can just sit at their desk and watch these programs and participate in them. And the reason we spend so much effort training scientists and training regulators is because we know it works. There are lots of methods out there that are non-animal that could replace animals, but they haven't yet because there are all these really difficult to define barriers, like lack of training, lack of understanding of what the non-animal methods can do. Regulator comfort is sometimes a term that is used to express lack of knowledge or understanding, and regulations that are in place and that kind of prevent the implementation of new methods. So these training sessions that we hold allow people to talk about those barriers, to learn about the non-animal methods. And we know based on surveys and based on experience that they do, once they've gone through trainings like this, they do accept or use the alternative methods.
And my hope would be that they also speak to their colleagues about the things that they've just learned. And if you do the math there, you're talking about, you said over 5,000, but if we just use the number 5,000, that's almost 14 researchers every single day that are learning about this. And that is just phenomenal to me. That is a great number. And I suspect that number will continue to climb into the new year. And to this end, you're talking about education and coaching up the researchers and the scientists. Well, talk to us about the success that you've had with our animals and education labs this year.
Yeah, so most people know that we, for a long time, have been working to replace the use of animals in medical training, different kinds of medical training, from medical school all the way to postgraduate residency training. And we have had a couple of victories this year that we're very excited about. We shut down Vanderbilt University's training lab, in which animals were cut open and killed to train doctors. We also worked with the University of British Columbia to stop their annual course from using animals before it even happened. And then this was the culmination of an almost 9-year campaign to get the University of Washington to stop using and killing pigs to train paramedics. So we've gotten all three of those switched over to human relevant methods, which is going to be helpful for their students, certainly.
But there we go. But it doesn't just stop with human relevant or human testing here, because you were also working to end a lot of this when it comes to veterinarians as well. We've had some success there. What has that been?
Well, people might be surprised to know that many veterinary schools, when you're training to be a veterinarian, there's something called a terminal surgery lab, where you learn to do surgeries on animals, and those animals are killed at the end of this training courses. And so we are one of the only groups working on this and trying to convince these veterinarian schools to switch over to to stop doing those terminal labs and use other ways to teach their students surgical procedures. So this year we been able to convince Tuskegee University and the University of Auburn, both of those veterinary schools have stopped doing their terminal surgery labs.
That's fantastic. That's fantastic.
It's great
and
news.
Yeah, and I'm guessing that there are others who hopefully will be following suit here in the future. I want to go back though to probably something that is more on the human side of things, as well as far as like human testing, and that would be toxicology tests. I know that specifically you and I have talked off camera about these dermal absorption tests. Had a big victory there as well. Talk to us about what those are and what we were able to do.
Well, this is very exciting for me in particular because I have been working on this for some time with a coalition of stakeholders. But so the dermal absorption test is a test that has been required in the past for pesticides. If a pesticide company wants to put a pesticide on the market, they need to know, is that pesticide going to be absorbed through the skin of, for example, workers in the fields? And so they would take 40 animals and put the pesticide on the animal's skin and then kill the animals to see where in their body and how much of the pesticide has been absorbed through the skin. But there has been an alternative available for some time. You can take donated skin from humans and create a little circular biopsy of that skin in the laboratory. You put the pesticide on that biopsy of skin and see how it absorbs. And then what you have is a measure of how the pesticide actually absorbs through human skin, which is the measurement that we want anyway. So we, a few years ago, we brought together all of the regulators and the pesticide companies and the test method developers to figure out, well, what are the barriers? Why is this test, which is available, which is better, not being used? And we talked through those. We published an analysis in the literature, and then there was another sort of analysis by some of the pesticide companies. And so this year, the EPA and Health Canada have started accepting the in vitro test alone. And we're saving about 400 animals every year with getting rid of that test.
That's fantastic. That is fantastic. And I want to end kind of on a big one that we have also talked a lot about at the Physicians Committee. This is something that our members have really gotten behind, and it's something that, for me personally, had flown way under the radar until you guys started talking about that. And that is the use of horseshoe crabs in the development of certain vaccines. And it never even dawned on me that a horseshoe crab could get wrapped up in all of this, but it's a major problem.
Yeah, it is. What they're used for is to test pyrogenicity. So we want to know, are vaccines and other injectable drugs, are they contaminated with bacteria that will cause a fever reaction if they're injected into patients? And this isn't just like most toxicity tests where they are conducted once at the beginning of the product marketing cycle. This is a test that's conducted on every lot. Every batch has to be tested to make sure that it's not contaminated. And sometimes ingredients that are going into the vaccines or the injectable drugs also have to be tested. So we're talking about billions of these tests every year. If you think about the coronavirus vaccine and how many of those are being produced. It used to be that this test was conducted in rabbits, and most of that testing has been replaced by this horseshoe crab based test. What they do is they go out into the ocean, they capture horseshoe crabs, they bring them to a laboratory, and they take some of their blood, and then they take them back out to the ocean and release them. And this is touted as an alternative because it's not using a live rabbit, it's only using the horseshoe crab's blood, but you have to get that blood from those animals. And of course, it causes them trauma and pain and suffering. And we know that a certain percentage of the horseshoe crabs do not live. They're released back into the ocean and die. So these animals are fascinating animals. They've been around since before the dinosaurs. And so it's really a shame that we've now turned them into this blood factory in order to test the vaccines, especially because we have a laboratory produced recombinant that can do the same thing as the material in the blood can assess the vaccines for pyrogen contamination. So what we're doing is we're working with the major vaccine companies and pharmaceutical companies together in a coalition. And we're working to convince the Food and Drug Administration and another agency that people probably aren't aware of, the US Pharmacopoeia, which both these agencies set these standards for these tests. So we're working together with them to push forward acceptance of the recombinant alternative. And we're making great progress. This year we had 2 sort of closed door meetings where we talk through all of the issues, just like with the dermal absorption. And we're working together with those companies to provide whatever data might be necessary in order to allow the regulatory agency to accept the alternatives. And I think in the next year, we're going to see that come to pass.
I've always been of the belief that when it comes to big business, big pharma, that money talks. Have you been able to do a cost analysis of these human relevant methods versus the traditional testing that we're seeing right now?
For pyrogenicity in particular, you mean, or in general?
Or just in general, generally speaking?
Yeah, I mean, it really depends, to be honest, on the test, because sometimes the tests are less expensive or they're faster, but sometimes there's a sort of a startup cost, if you will, because all their processes are within the current test and are kind of adapted to all of that. And so if they have to change over to a new test, there's some cost to that, to change their systems, to change their procedures and everything. But what's interesting with the pyrogenicity situation that we have found is when we talk to companies, they say, this is our ultimate goal, is to not use animals. And so we're willing to put in that extra upfront cost in order to get to that ultimate goal, because we see it as our corporate responsibility to avoid doing that.
Yeah. And I wonder, just like with a lot of other investments, you put that money up front, but then long term, you start to see this big return on said investment. And maybe that's where the cost savings come, in a number of years. But long term, I would think from a fiscal standpoint, it'd be a really beneficial thing. And then last point here is that when we're talking about these different methods of testing here, you are aware, and you brought to my attention something that is called animal model bias when researchers are trying to get their data published. What is that? What is the problem that's happening here?
Well, this is a huge concern because we know that and we've talked before about really advanced human-based methods like, the lung on a chip, or these really cool guts on a chip, or different models that are much better than using a mouse to model diabetes. But scientists' main way to be successful is to publish their findings in the scientific literature. So they submit their papers to journals and then they get published, and that's how other scientists find out about their work. And we have started to hear kind of rumors that scientists who use in vitro models, human-based methods, were having a hard time publishing their work. And we're even potentially being asked to do animal experiments in order to demonstrate that the data that they found with their human-based method was right, which doesn't really make any sense at all. So we decided to look into this. And so our team has been working with another organization to do a big survey of scientists to see whether there was any truth to these rumors. And so we've got the paper the survey results, we're analyzing them, and we're about to put them in a paper of our own in the scientific literature to call attention to this problem. We definitely found that scientists are asked by the journal editors and reviewers to validate their findings in animal models, which is not what they should have to be doing, because that's animals that are killed for no reason. And it also perpetuates this idea that animal experiments are better than human-based in vitro methods, which we know is not the case.
All right. And last but not least, made a lot of progress this past year with schools in particular. Looking ahead to the new year, 2022, give me a cliffhanger. How many schools right now are in your sights?
Well, we are going to get all of them eventually, but we've got about 6 of them that we're really actively campaigning to right now, trying to get them to replace their animal training programs. So we have to be a little opportunistic. Sometimes, our plans are such as they are, and then something comes up such that we'll be able to address another school. But yeah, we've got about 6 medical schools in our sites. We'll see what we'll be able to achieve. Of course, with PCRM supporters are always behind us. We know we will win, and we'll just have to see.
Yeah, stay tuned. 2022, it's going to be an exciting year. Lots of work still to do, but I have no doubt that you and your team are going to accomplish even more great things. Kristie Sullivan, Vice President of Research Policy, thank you so very much for joining us, and Happy New Year.
Thanks, and same to you. It's great to be here.
If your health IQ is a couple of points higher than it was a few minutes ago, go ahead and like this video or subscribe to the YouTube channel. And to take it even higher, head over to Apple Podcast or wherever you get your favorite shows. Look for The Exam Room by the Physicians Committee. Hit the subscribe button there as well and help to make your world a healthier place.